Welcome, Brookings AP Bio students. This is Mrs. Rydell, and in this slideshow, we're going to look a little closer at what are some of the ways that we can use the information we know about DNA to create useful tools for society, and that involves technology, what we call biotechnology. This is a slideshow that's been modified from one that came from Kim Foglia's amazing site. We're going to look a little bit about something called genetic engineering. How can we manipulate DNA? How can we use the DNA, the information that we know about DNA, to engineer, to take genes from different organisms and use them in ways that are beneficial to humans? And in order to do that, to manipulate genes, we're going to need some tools. And so we're going to talk about a couple different tools that are used in that are part of our toolkit for manipulating genes. If you think about ways that we use DNA, using fingerprinting for crime scenes, creating medicines for humans, engineering organisms for food, or looking at genetic disorders and gene therapies, and using it to analyze chromosomes and see evolutionary sequences, creating new organisms. All of these are ways that we use technology and our information that we know about DNA for useful purposes. So, what are some of these technologies? First of all, we can use technologies to help us diagnose disease, detect viruses, look at humans and see if they're carrying genetic mutations for genetic disorders. Look at gene therapy. Once we ID that a person has a genetic disorder, has a certain gene, we can isolate those genes. We can use different kinds of therapy methods to create help for people who have genetic disorders. We use DNA technologies in making pharmaceuticals. Right now, people who have diabetes, the insulin that's produced to help them is grown in bacteria. We have bacteria producing all kinds of human proteins, human growth hormone, insulin. We use it in crime scene analysis. We use it in creating new organisms. And so all of these are ways that we use what we know about DNA and create new technologies for the benefit, hopefully, of humans. One of the big topics, and we're going to talk a little bit more about this, is using genetic technologies to create what we call genetically modified organisms, GMOs. You may hear that phrase. And what we're doing is combining genes, moving genes from one organism to another to create a genetically modified organism that has new genetic traits that it didn't have before. A good example is BT corn. BT corn is corn that has a gene incorporated in it that allows, that came from a bacteria, and allows the corn to have kind of its own built-in pesticide. It creates its own toxin that kills the caterpillars that would eat the corn, and it's built in to the corn plant by transferring a gene from a bacteria. We've moved lots of genes in different organisms for creating and helping us to grow more food on the planet, and it may be a possibility for allowing us to provide more food for the growing population that we have. Being able to extend growing season, for example. Strawberries that have received a gene for prevent them from freezing and allow them to have a longer growing season, or grow in places where they couldn't grow before. Improving the quality of food. Golden rice is a form of rice that has an artificially introduced gene for making vitamin A, in improving the nutritional value of the rice. All of these are what we call genetically modified organisms, and we're going to talk a little bit more about the pros and cons of these. So, one of the tools that we use to create these genetically modified organisms, or to move genes around from one organism to another, is the little ring of DNA that we talked about back in our bacteria. We talked a little bit about bacteria genetics, and it has to do with plasmids. Remember, plasmids are small circular loops of DNA. They're extra chromosomal. They're in addition to the bacterial chromosome. And we are going to use plasmids now as what we call vectors. Vectors are carriers for genes that we're introducing into another organism. And so if we take the plasmid, add the gene that we want to move, and then introduce it into the bacteria, into another organism, it's now going to allow the bacteria to pick up new genes and express those genes, make new proteins that they couldn't make before. So we're going to take the genes from the two different organisms, we're going to glue them together, and then use that recombinant plasmid as a vector to transport our gene that we want into our new organism and transform the bacteria, allow it to express those new genes that it has acquired. So we take the gene we want, for example, insulin, human growth hormone, lactase for people who are lactose intolerant, and we can create these what we call recombinant plasmids by cutting the DNA and cutting the plasmid and then gluing them together, inserting the gene that we want, and then we have a vector to move that gene into another organism. And the glue we use is ligase. We had that enzyme, then we talked about DNA replication. We're going to use some of the information now that we know about how DNA works to help us with these new technologies. So how are we going to cut our DNA? We're going to use something, some enzymes called restriction enzymes, short for restriction endonucleases. And these were discovered back in the 1960s, and they evolved in bacteria to help protect them against foreign invading DNA. Remember, viruses inject their DNA into a bacteria cell, and so bacteria evolved these enzymes to help protect them against invading viruses. So if DNA should get into their cell, and the cell recognizes that as foreign DNA that's not supposed to be here, these enzymes then would take the DNA and chop it into pieces to prevent the virus from infecting the cell. We have found these in bacteria and taken advantage of them now by their ability to cut DNA. And so let's take a look at how that's going to work. What do you notice about these phrases? If you take a look, what you should notice about them is they read the same backwards and forwards. R-A-D-A-R, R-A-D-A-R. W-A-S-I-T-A, W-A-S-I-T-A. You can see the phrases, the sentences, read the same. They're called palindromes. Palindromes are phrases that read the same backwards and forwards. And the interesting thing about restriction enzymes is they always cut the DNA at specific palindrome sequences. They do a snip in the DNA and they cut asymmetrically. So we're going to cut our DNA. If this is my little piece of DNA, those restriction enzymes look for specific sequences, and different enzymes cut at different sequences. They're called restriction sites, the place where they're going to cut. And oftentimes they are palindromes, G-A-A-T-T-C, G-A-A-T-T-C. They read the same forwards and backwards on opposite sides of the strand. And when we cut now, we're going to cut unequally. We're not going to cut just straight across. It's going to make a little kind of a zigzaggy cut. And so what it does is create what we call sticky ends. These ends now on my piece of DNA are going to allow them to be able to bind to another piece of DNA that's cut with the same enzyme that has the same sticky end, because remember, we have matching base pairs here. Lots of restriction enzymes have been found from lots of different organisms, different kinds of bacteria. Usually they're named after the organism they're found. EcoR1 comes from E. coli, and this was the restriction enzyme, the first restriction enzyme that was discovered. Since that time, we found lots of different Hindi3, BAMH1. There's lots of different enzymes that can be used to cut DNA. They're kind of like magic scissors that can cut the DNA in different places, in different sequences. So we're going to create these pieces of DNA that have what we call these sticky ends, and then use those sticky ends to combine bacterial DNA, plasmid DNA, other kinds of DNA from different organisms, and create these recombinant plasmids. The discovery of restriction enzymes, like I said, happened in the 1960s. These are the gentlemen who discovered them. They did receive the Nobel Prize in 1978. And if you look now, we're gonna use these restriction enzymes to cut the DNA in these zigzaggy sticky end, leave these sticky ends on the ends of the DNA, and allow the technology to be used for recombining DNA from different organisms. So if you look at restriction endonucleases, restriction enzymes, and how they work, each enzyme is going to cut the same piece of DNA in a little different way, depending on what sequence it recognizes. So different kinds of DNA cut with the same enzyme are going to have the same kinds of sticky ends. We're going to watch a little video about restriction endonucleases.
Restriction endonucleases are enzymes that cleave DNA at specific nucleotide sequences. The sequence recognized is often 4 to 6 nucleotides long. For example, the restriction endonuclease EcoR1 recognizes the sequence GAATTC. The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The two strands of the DNA duplex have the same nucleotide sequence running in opposite directions for the length of the recognition sequence. Because the same recognition sequence occurs in both strands of the DNA duplex, the restriction endonuclease can bind to and cleave both strands of the DNA molecule. Because the bond cleaved is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end a few nucleotides long, and the single-stranded ends of the two fragments are complementary to each other. These single-stranded ends can pair with each other, sticky ends. Once their ends have paired, the two fragments can be joined together with the enzyme DNA ligase, which reforms the phosphodiester bonds of DNA. What makes restriction endonucleases so valuable is that any two fragments of DNA produced by the same restriction endonuclease can be joined together. Restriction endonucleases are fundamental tools in genetic engineering.
If I cut at that recognition site, it's going to leave these kind of a zigzaggy cut, are going to leave these sticky ends, and allows DNA from different organisms, if it's cut with the same restriction enzyme, to have that same matching sticky end and allow us to combine genetic material from different organisms. So if I'm going to take two pieces of DNA here, this is the gene that I want. Maybe it's the end of an insulin gene here, or a human growth hormone gene, or a gene for making vitamin A that we're going to move into golden rice, or any gene that we want, and we're going to take this piece of DNA that's the chromosome that we want to add it to, and if I cut it with the same sticky ends now, I can create a recombinant piece of DNA. It has DNA from two different organisms combined together. Why would we want to do this? We've done this for creating medicines, pharmaceuticals, by combining human genes, for example, the human insulin gene, and putting it into a bacteria. And the bacteria now, remember, we're sharing the same genetic code. The bacteria can use the information in that gene to create insulin, and we can grow bacteria and make insulin outside the human body to create medicines for people. So here's my bacteria. I'm going to use it to make medicine. Human insulin, using the human insulin gene, can create human insulin for humans. And it's because bacteria use the same gene code. Once we have the code there, they can use their ribosomes to translate the message and make human products. Remember that code is universal. All living organisms, they use the same DNA. We're sharing the same language. We're reading genes in the same way. We're using the same amino acids to build our proteins. And so bacteria are unique organisms that can be used to create human products. We've made our little plasmid now. So this is a recombinant plasmid that has our genes that we have added to the plasmid, and now we need to get our bacteria to take up the plasmid. Remember, bacteria are very good at picking up, that's that transformation, picking up naked DNA that are introduced into their environment. And then once we get the plasmid into the bacteria, we can grow those bacteria in culture, and the plasmid will be copied with that gene along with the bacterial DNA when the bacteria grow and copy itself, and make lots of bacteria that have copies of that plasmid. That's called cloning the plasmid. And we're going to make copies of the plasmid along with that gene that's been inserted, and create a whole population of bacteria that can create this new protein that has the insulin. So remember that sequence. DNA makes the RNA message, makes the protein. And even if the message is coming from another organism, it can still be produced and turned into a protein like insulin. So we're going to take our plasmid, we're going to snip, we're going to combine it with the genes from another organism and make that recombinant plasmid. We're going to use that as a vector to transfer into our bacteria. They're going to pick up that new DNA transformation, and we're going to use it to create a whole population of bacteria that contain that recombinant plasmid, and then use that to make insulin for us. So how do we get rid of the junk? Remember, eukaryotic organisms have a lot of extra DNA in their code that does not end up in the final protein, in the final message. There's processing, there's editing of those introns that take place in the nucleus. So if I want to move the gene that's going to have the final information in it, I need a piece of DNA to go into my plasmid that does not have those extra introns. Remember, introns, bacteria don't do processing. They don't edit out those introns. So what I need is a copy of the gene that does not have the introns. And that's what I need to put into my bacteria cell, that I need to put into my vector. So we're going to use, if we use that message, that has already been edited. But how do I get now from my edited message, turn it back into a piece of DNA that has the edited stuff removed? I'm going to use another trick from the viruses. We can't use our DNA directly, right? We're going to use the DNA to make a message. And then we're going to use an enzyme. Remember we talked about viruses, retroviruses. They have an enzyme that allows them to take their genetic RNA code molecule. Remember, some viruses have their genetic code as RNA, not DNA, like the HIV virus. We're going to take that RNA code and turn it into a piece of DNA by using an enzyme called reverse transcriptase. It's found in retroviruses. And we can take now that message that's already been edited, remove the introns, and turn it into a piece of DNA that does not need to be edited before it is used to transcribe a message and make our protein. Reverse transcriptase is found in retroviruses. Remember, those are the ones that have RNA for their genetic code molecule, not DNA. And we're going to use that RNA message from our human cell to turn, go backwards and turn it into a piece of DNA. And so in this case, really, we're kind of contradicting that central dogma of how information passes in cells. In this, we're taking information from an RNA molecule and turning it back into a piece of DNA. We're going to take our eukaryotic RNA message after the introns have been removed and turn it back into a sequence of DNA that can be read directly by the bacteria without processing. Here's how this works. If this is my message, messenger RNA, we're going to use transcriptase to start with the message and reverse engineer the matching strand of DNA, copy it off of the RNA message. And then once I get this little DNA strand that matches the RNA, I can use that to make the matching, remember DNA is double-stranded, the double-stranded DNA match to create a piece of DNA that codes for the information for making this RNA message. It's called C, or complementary DNA. Once I have my plasmid with the gene that I want transferred into a bacteria, I need a way to select for those bacteria that contain the information, that contain the new gene. And so one of the ways that we do select for a bacteria containing the transfer gene is to also include on the vector, on the plasmid, genes for antibiotic resistance. Remember, plasmids can carry those antibiotic resistance genes. So antibiotics become a selective agent. If you grow a plate out bacteria on a plate that has antibiotics on it, only the bacteria that contain that recombinant plasmid with the antibiotic resistance genes are going to be able to grow on the plate. So it's a way to select for those bacteria in the population that have picked up the recombinant plasmid. So if you grow them in normal media, all the bacteria can grow. If you grow them on media that contains the antibiotic ampicillin, only the bacteria that have the transformation happening, only the transformed bacteria that contain that antibiotic resistance gene are going to be able to grow. And basically what we're doing is cloning. We're creating a population of organisms that contain the gene that we're looking for. Another way we can detect whether organisms have picked up a plasmid is by adding a gene that we can produces a protein that we can see. And jellyfish create something called GFP, green fluorescent protein. There's certain kinds of jellyfish that make this protein that causes them to glow in this weird fluorescent green way. And we have used that gene as well, instead of the antibiotic resistance gene, taking that gene from the jellyfish, incorporating it into a plasmid, and then putting that plasmid into bacteria. And the bacteria that have picked up the plasmid are going to be able to glow. We've moved that plasmid not only into bacteria, but lots of other organisms, including mammals. We've created GFP mice, GFP pigs, GFP monkeys. That green fluorescent protein, the GFP, is a genetic tool. Originally it came from jellyfish, and if we have an organism that has incorporated that glowy gene, it's a way to tell it apart from organisms that have not picked up the plasmid. They're not going to glow. And if it has the glowy gene, that we know that it also is carrying the other gene that we're looking for. This is kind of like a word processing program. We need a way to cut the code. That's the restriction enzymes. We can paste information into our Word document by using ligase. We have a way to copy information by putting it into a plasmid and growing it into a bacteria, cloning it with using the bacteria to create more bacteria that have the recombinant plasmids. But there might be an easier way. That takes a lot of time in moving DNA into a bacteria cell, getting them to pick up those plasmids. There is another way that we have come up with to make multiple copies of a piece of DNA. And first of all, we have to find the piece of DNA that we want to copy. But then how can we maybe do this in a little easier way? And that is what's coming in our next part of our slideshow, which is looking at other ways to manipulate DNA. So stay tuned for our second part of the slideshow, and then after you have watched that, you're going to take your little video quiz.